So good morning, my name is Shaliq Hassan and I'll be presenting a preclinical study on gefapixant, also known as ERISA. It's an EGFR tyrosine kinase inhibitor. EGFR or epidermal growth factor receptor is a protein which is present on the surface of the cells that receives growth signals and transmits them inside the cell. So it is important because it controls a normal cell growth, survival, and differentiation. But when it overacts or mutates, it can lead to cancers such as lung cancer, colorectal, or breast cancer. So EGFR can be detected by using immunohistochemistry, fluorescence in situ hybridization, and PCR, which helps in detecting the protein expression, gene amplification, and mutations. And while detecting, this can be a important target for therapy with including drugs like tyrosine kinase inhibitors such as gefapixant or monoclonal antibodies like cetuximab. So Irisa is an targeted anti-cancer drug that belongs to the class of tyrosine kinase inhibitors, and it is mainly used for treatment of non-small cell lung cancer, and it's particularly seen in patients whose tumor carries activating mutations in EGFR. And it was discovered by AstraZeneca and as the first smallest molecule of EGFR tyrosine kinase. So during the development and discovery phase, numerous essays and studies were conducted on ERISA to test the compounds for the EGFR inhibition of autophosphorylation and to check the specificity, potency, and oral availability of the drug. And during preclinical development, in vitro cell proliferation assays, apoptosis assays, and combination assays were conducted to check if the ERISA induced apoptosis and to test the synergy with the chemotherapeutic drugs. And in vivo xenograft tumor growth assays and survival assays and toxicity monitoring were used to demonstrate efficacy and to test the therapeutic effect of the drug. So in this preclinical study, the aim of the study was to test the anti-tumor effect of the ERISA alone and to evaluate the synergy with chemotherapy. And the methods were used in vitro and in vivo, where in vivo we used GEO colon cancer xenografts in nude mice, and in vitro we had ovarian, breast, and colon cancer cell lines. So while moving on to the result, in vivo, experiments with GEO colon cancer xenograft showed dose-dependent activity. So in figure A, from the graph, we can see that the tumor growth was partially inhibited at 1.25 and 2.5 mg dose per day, and while administering 5 mg dose per day, tumor growth was almost completely suppressed, and the effect was cytostatic. So as the that the treatment stopped, the tumor growth was resumed. And in case of figure B, survival curves, in control group, all the mice died within 6 weeks, and at 5 mg per day, survival was significantly prolonged, with many mice surviving much longer. So in the slide results, we can see that gefapixant was combined with chemotherapeutic drugs such as paclitaxel, cisplatin, etc. And the combination was almost completely suppressed tumor growth during the treatment, and the tumors regrew only after 4 to 8 weeks. So the survival curve shows that the strongest benefit with paclitaxel plus gefapixant combination, which shows around 40 to 50% of the mice were still alive at 16 weeks, rather than compared to the 6 to 8 weeks for single agent groups. So this highlights the high synergistic activity of gefapixant with cytotoxic chemotherapy. So this is the flowchart and the data compiled from the experiments results. So in this previous results, we could see that the ERISA alone had a cytostatic effect, but with combined with chemotherapeutic drugs or chemotherapy, we can see a cytotoxic effect. And gefapixant was having a high synergistic effect with paclitaxel, and it had a synergistic effect with all the drugs, but it was showing more effect with paclitaxel, which was 50 to 40% of the mice were surviving 16 weeks. So it was more having a better therapeutic effect than the other drugs. So from these preclinical trials, we could see that not all patients are benefited from EGFR TKI inhibitors, And a real turning point came in 2004 when EGFR mutations were found in non-small cell lung cancer, which led to the molecular screening, where only mutation positive patients were given EGFR TKIs. So from these benefits were very clear because it had higher response rates, and it had a better quality of life because TKIs were oral and it was less toxic. So over time, resistant mutations like T790M were managed with newer drugs such as afatinib or osimertinib, which had a better outcome compared to the other drugs. And this introduced a genotype guided precision therapy, which managed to improve the patient outcomes dramatically. Thank you.